Hello. We are now going to be moving into discussing cells. So we're done with protein synthesis for now. We will be talking about it again and referencing it as we go forward. However, we're done with that for now, but we still need to look at cells. What is a cell? What are the type of cells? What are the parts of cells so that you can better understand how the future units are going to go. So I first want to start off by discussing cell theory. And cell theory states a few different things. First, it states that all living things are made up of one or more cells. So if you are a living thing, you are made up of at least one cell. Two, cells are the basic unit of life. So that kind of ties in with the first thing. So if you are living, you are made of cells. And number three, all cells come from pre-existing cells. So this one is kind of a chicken and egg situation, if you think about it. The whole which came first, the chicken or the egg. Obviously all the current cells that exist came from cells that existed before. But if you kind of think all the way back to what about the first cell? If it's the first cell, obviously it didn't come from another cell. But there have been studies done about where cells came from, how did cells evolve, what came before them, blah, 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 blah. One experiment that you may have heard of, maybe not, but I first learned about it in middle school, is the Miller-Urey experiment. And what happened was these scientists basically took all of the elements that were available on way, way, way prehistoric Earth, and they put them all together in a closed system, and they applied some energy to that closed system. And the elements, the molecules that they were including were things that were available back then. So like carbon dioxide, like methane, like nitrogen, a couple of other things. And they left it alone for quite a while. And then they looked back later, and what they found was that instead of just those very simplistic molecules that they had started with, now they were seeing much more complex things that were precursors of complicated organic molecules that we've actually studied. So things like the precursors to proteins or the precursors to nucleic acids, building blocks that could later be made up into those things. And that's really, really important because that means that organic molecules, the things that are necessary for life, could have popped up without the need for other life before. So basically, what I'm telling you is that experiment proves that the precursors for life could have formed at that time on their own, completely spontaneously. So let's continue on. Let's talk about what a cell is. Well, the definition of it is the smallest structural and functional unit of an organism, or the smallest unit of life. So to be considered a cell, something has to have a couple of things. It has to have genetic material in it. And genetic material we're defining as either DNA or RNA. It has to be enclosed by some sort of membrane. And it must meet the Go-Racer criteria for life. So what is that? Things that are alive must be able to grow and develop. They must be organized. They must be able to reproduce. They have to be able to adapt through evolution. They have to be made of cells. They have to be able to use energy, and they have to respond to stimuli. So I'm going to talk about that a little bit more later in this presentation, but those are the qualifications of life that we're going to use. These other images here, the one on the far left should look familiar to you if you've ever seen the movie Jurassic Park. This here in the middle is the traditional DNA image that we've seen a million times. And then on the top right, there's a little cartoon image of a cell. So that purple line that you're seeing around the outside here, that is representative of that membrane that cells have to have, and then everything inside the middle section here should be that genetic material that cells have to have. So again, though, that's a pretty broad definition. If all you need to do to be considered a cell is have a membrane, have genetic material, and do those things, there's got to be a crap ton of cells, and there are. So this image here with all of these types of cells listed, those are just a couple of all the types of cells that exist in your body. Humans have 37 trillion some odd cells of 200 different types, but that's excluding every other type of life that exists. So if we're just talking about humans, what about certain animals? What about plants? What about fungi? What about bacteria? What about all of those other things? So there are a bajillion different types of cells, which is a lot. So we define cells, or we split cells, I should say, into two main categories: prokaryotic cells and eukaryotic cells. Now, we are eukaryotic organisms. We have cells that mimic this one on the right, because we have cells that have nuclei. We have cells that have all of this stuff in the middle. Prokaryotes, on the other hand, otherwise known as bacteria, they don't have a whole lot going on for them. This purple stringy stuff that you're seeing in the middle, that's their genetic material. And these little tiny dots that you're seeing are ribosomes that allow the bacterial cells to make proteins. But other than that, they don't really have all the other things that you see in the eukaryotic cells. Another thing that this diagram does not really do a good job of representing is the size difference between a eukaryote and a prokaryote. Now, are there some very small eukaryotes? Yes, there are, absolutely. But on the whole, a bacterial cell compared to a human cell is going to be like this, maybe. So this little thing that I just colored right here, that's the comparison between how big this thing actually is compared to a human eukaryotic cell. So talking about prokaryotes, I love this image. That's the background image, because bacteria always look so funky and alien to me. A definition of a prokaryote is cells that do not have a nucleus and no organelles with membranes. So they do not have membrane-bound organelles. But the biggest thing that I want you to know about bacteria and prokaryotes is that they are the same thing. If I say the word prokaryote, I'm talking about a bacteria. If I say the word bacteria, I am talking about a prokaryote. Eukaryotes, on the other hand, they do contain those membrane-bound organelles, like I was saying before. This image over here is a scanning electron microscopy image of a eukaryotic cell. And in it, you can see some really compelling things. This big, big, big dark spot right here is the nucleus, where all the genetic material is. These things over here, this here, this, all of those are mitochondria. That's where that energy production is. This little light spot in this one and this one over here that you can see, those are lysosomes that are going to function to break down old proteins or other things like that. The plasma membrane is existing around the outside, so basically the barrier between the inside and the outside of the cell. We can kind of see a shadowy, rough endoplasmic reticulum over here. There's a lot of stuff in eukaryotic cells, basically. And eukaryotic cells have different subtypes as well. Plant eukaryotic cells are going to be a little different than animal eukaryotic cells, which will be different than fungal eukaryotic cells, and protists as well. So I need to back up for a second. I need to define the word organelle, because I'm sure that you have heard the word organelle before, mostly because I have said it several times so far and in class this year. But what it is specifically is a specialized structure within a cell that floats around in the cytoplasm, in the liquid part that exists within cells. So you can think of your own organs and how each one of your own organs has a specific structure and a specific function within your body. Organelles are the same. They have specific structures and specific functions within cells. So this giant list right here is a list of the ones that we are going to talk about. Are there more than this? Yes, absolutely. There's always more. But I'm not going to talk about every single organelle that could possibly exist within a cell. We'd be here for a long time. I'm hopeful that you have at least heard of these before, because you should have studied them, and I think it was 6th grade at this point. You should have studied cell parts already. You should be at least somewhat familiar with this information. Here is a cartoon-like image of some of the different organelles: Golgi apparatus, smooth and rough endoplasmic reticulum, the ribosomes, the nucleus, etc., etc. Plant cells and animal cells have a lot in common. They both have DNA, they both have ribosomes, cytoplasm, a cell membrane, a cytoskeleton, mitochondria, Golgi, an endoplasmic reticulum, a nucleus, and a nucleolus. But they do have some differences as well. Animal cells can be so many different shapes. They aren't constrained by a cell wall that makes plant cells be very boxy. Can plant cells have different shapes? Yes, absolutely. But they're very boxy. They're very geometric. They can be like hexagons or rectangles or pentagons or things like that. But they don't have as much variety as animal cells do. And as we look at different types of animal cells going forward, you will see how different some animal cells can be. Plant cells also have chloroplasts. You should know what a chloroplast is by this point. Animal cells do not have chloroplasts, mostly. There are a few different animals that can actually do photosynthesis. There's a sea slug that can do it, Elysia chlorotica. There's a salamander that can do it, and there's aphids that can do photosynthesis and have the ability to absorb chloroplasts from their food and then repurpose them. But that is very rare. So on the whole, only plants have chloroplasts. Plants also have this thing called a central vacuole, which is used for water and nutrient storage. So if you've ever seen lettuce wilt before, that is happening because the vacuole in that cell is losing some of the water, because that vacuole acts not only as water storage because the plant needs water, but also structural support. It's not all the cell wall that gives plant cells their rigid shape. It's the vacuole as well. And then down here, obviously, plant cells are usually boxy or square-ish in shape. Here's a quick diagram of the anatomy of an animal cell. You'll see that that animal cell is kind of rounded. Not all animal cells are round, as I had expressed before, but this is a generic diagram. Here is the generic diagram of what a plant cell looks like. You can see in it this big, big, big shape that I'm tracing poorly right here is that central vacuole, which is going to contain a bunch of water. You'll see the chloroplasts over here they have those green discs in it. And of course, the outline of the cell wall. We got to talk about single-celled organisms and multicellular organisms as well. So some things are only made of one cell. Bacteria is probably the easiest example of that, but there are others as well. Not all single-celled organisms are prokaryotic. They can be eukaryotic too. Examples that you may have heard of would be amoebas, paramecium, caudatum, yeast, streptococcus. All of those would be examples of eukaryotic. Sorry, not all of those. Amoebas and paramecium would be examples of eukaryotic single-celled organisms. Streptococcus is a bacteria. My apologies. Multicellular organisms are things that you are guaranteed to have heard of before, because plants, animals, and humans, even though we technically fall under the kingdom Animalia, are multicellular eukaryotes. Multicellular organisms, though, because they are so big and so complex, their cells have to differentiate into different types of cells with different functions within your body or the plant's entire structure. Real quick though, before I move on to that, this over here and this over here those are two examples of eukaryotic single-celled organisms. And you can tell they're eukaryotes because you can see a bunch of stuff in them. You can see a bunch of membrane-bound organelles happening, whereas prokaryotes, in this image, this is Streptococcus over here, they're very tiny and you can't see anything within that cell. There's no structure there that you can visibly see. So here's a nice additional diagram of different types of cells that exist in your body. Again, there's more than this, but you can see how different some of them look for one another. And this kind of goes to exemplify what I was saying about animal cells being able to be so many different shapes, because we have a sperm cell here that has a giant tail on it. We have neurons and bone cells and things like that. Like, here's our neurons over here that are super long and branching. Here's our bone cells that look kind of awkward over there. Here's some really, really long cells with more than one nuclei in it. Boxy cells, round cells. There's a ton of different shapes that you're seeing because those cells have a ton of different functions. So diversity of form reflects diversity of function. And what that means really is that if a cell has a different job, it's probably going to look a little different from a different type of cell. And the best example I could come up with that exemplifies that would be a nerve cell. Because think about it, a nerve cell's job is to send messages from one place to another. So it wouldn't make any sense for that cell to be small and round, because then it can't send messages over long distances. So instead, neurons are long, they're branching, they have giant axons to send messages to other places. But cells all in your body all have the exact same genetic code in them, with the exception of sperm or egg cells, because again, they have half. But not all genes are on all the time. Some genes are switched on when necessary, and that's why your cells with the exact same genome can look so different from one another. All your cells start out as stem cells, which are undifferentiated cells that haven't become what they're supposed to be yet. So a stem cell could become a neuron, it could become an enterocyte that goes in your intestines, it could become a liver cell, it could become a heart cell, it could become a bone cell, it could become whatever, which is why stem cells are so compelling in scientific research. Because if you have a Parkinson's patient or an Alzheimer's patient with a bunch of neurons that are degenerating and not working right, if you could treat that, if you could use stem cells to kind of push them towards differentiating into neurons and then have new neurons that aren't damaged yet, that could be a very groundbreaking treatment for Alzheimer's, for Parkinson's, for horrible, horrible diseases that wreak havoc on so many people. So that's part of the reason why stem cells are really important. Last thing I really want to discuss is viruses, because viruses are very important to understand, particularly now since we are in the middle of a viral pandemic. What a virus is an infectious agent that's tiny, and it's very simple, and it can multiply, but only on living cells of plants, animals, or bacteria. So down here, we have a couple of examples of different types of viruses. The one on the left that you're seeing here and here, those are phages, bacteriophages. And they contain all their genetic material in this little portion that I'm circling right now. And they kind of latch onto a cell and inject that genetic material in. The coronavirus, which you're seeing here and here, looks very, very different. It was so named because of the spike proteins that over here you see in yellow. Looking at that under a microscope, they thought that those spike proteins looked kind of like a little crown, because corona means crown, and I forget what language, to be honest, but I think it was either Latin or Greek or something like that. It means crown, and so they named the coronavirus after the little halo crown situation that they were seeing. We do not consider viruses to be alive. Why is that, though? And to answer that question, I need to go back to that Go Racer qualification of life situation that I was talking about earlier. So do viruses do all of those things? And the answer is no. Do they maintain homeostasis? Homeostasis, by the way, if you've never heard that term, is essentially a balance that living organisms maintain within themselves. So like for us, we have to maintain a certain temperature range, we have to maintain a certain pH range, we have to maintain a bunch of things in our body. So if you are getting too hot, your body will sweat to cool you down, things like that. But viruses do not maintain internal homeostasis. They kind of exist in a suspended animation state until they come across a host cell, in which case they replicate themselves or get the host cell to replicate for them, rather. Do they have levels of organization? Yes, they do. There are different kinds of viruses. They're organized differently. So that one, viruses do. Do they reproduce? Kind of. I mean, they need a host cell to do it. So that would be like the same thing as if we couldn't produce our own young and we had to use a host to do it for us. So not really. Do they grow? No, they don't, because viruses are produced and pop into being exactly as they will always be. They don't grow, they don't change in that sense. Like it's not as if a host cell produces a little baby virus, it's going to grow up into an adult virus. No, the virus is produced as it is. Do they use energy? Not really. Again, they kind of just exist in suspended animation until their replication is triggered. So no, not so much. Do they respond to stimuli? Kind of. Their stimuli that they respond to is basically contact with a host cell, but other than that, not really. Do they adapt to their environment? That one, yes, for sure. Viruses can mutate. They do it all the time. That's why there is a flu vaccine every year, because there's so many different strains of the flu because of mutations. So that is why we don't consider viruses to be alive, because they don't do all of those things. And then I just wanted to really quickly show you some cool stuff, i.e. photos of cells that I took when I was an undergrad. So this first couple of images, this is a cocci bacteria, and we gram stained it, and we took pictures under a generic light microscope. So what you're seeing here and here, they are clusters. So I zoomed it in over here so you could see, but there's basically 1, 2, 3, 4, 5 cocci bacteria there. There's 4 in this cluster over here. This one is only a single cocci bacteria, and you can see instances of that over here as well, just single ones. But these are tiny, 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 tiny little bacteria. Next, we have a transmission electron microscopy image of a T4 bacteriophage. So those are the ones that look like little kind of spiders. This is where all the genetic material would be kept. This is that little body on it. The legs should be here, kind of coming off and branching off. They're not, because a lot of times when you take images of tiny, tiny virus particles, those legs are very fragile, and so they break off a lot, especially when you've never taken a TEM image before. Like when I prepared this sample, it was the first time that I'd ever done it. So we got an image, but I would have been very lucky if the legs had managed to stay attached. Here is a scanning electron microscopy image of a mouse fibroblast. So what you're looking at here is actually pieces of the cytoskeleton and the membrane of those mouse fibroblasts. So it looks very, very rough, because a cell membrane, while it seems like it should be smooth, it's not. A lot of times it's very, very rough looking. And then finally, we have some fluorescence microscopy images. Fluorescence microscopy is one of my favorite things. to do with microscopy because it's so beautiful if you do it right. So what happens is you take these different dyes that are going to bind to different things in the cell. So here, the blue, the light blue that you're seeing pop up like here and here, that dye bonded onto the genetic material of the cell. So what you're seeing pop up in light blue is the nuclei of all the different cells. And the red that you're kind of seeing in the background is bonded to the cytoskeleton of these mouse fibroblasts. So you're seeing the actual cytoskeleton light up. And the way that that works is once you expose the sample to the dye, it'll bind on where it's supposed to bind on. And then what you do is you expose it to different wavelengths of light. And because it's fluorescent, when you expose it to those wavelengths, it will glow. So we took, essentially, to sum up, glow-in-the-dark images of these cells. And so here's a little bit less bright image so that you can see the cytoskeleton better. But fluorescence microscopy is a ton of fun. And then these images here were taken without dyes. So what I did with these was I took very, very thin layers of a lily leaf and a lily petal, and I just stuck them under the microscope just to see what would happen. And actually it turned out really, really well because you can see the edges of all of these different cells in this image over here. And then over here that zoomed in farther, all this green stuff that you can see, all these green dots, those are chloroplasts. And they popped up really well because chloroplasts have a natural green color to them, so you can see them under the microscope. Most of the other organelles don't really have that, and so they're harder to see. And then the petal over here obviously has some color to it, so you can see those cell walls very, very easily. So hopefully you were able to fill out everything necessary from this presentation. Please ask me if you have any questions.